Good morning, students. In today's class, we learn about Lac Operon. So, before we look into the Lac Operon, we should know that it is nothing but a kind of gene expression which is found in prokaryotes. The control of gene expression. Each cell in the human contains all the genetic material for the growth and development of a human. Likewise, if you look into any organism, each cell is responsible for growth and development of that kind. So some of these genes will be need to be expressed all the time. These are the genes that are involved in vital biochemical processes such as respiration and all housekeeping metabolism. Other genes are not expressed all the time. They are switched on and off at need. So if you look into gene expression, there are two kinds of gene expression. One is said to be the constitutive gene expression, where you can see genes express continuously without any intermitting process. Whereas in another type of gene expression, you can see genes are switched on, off at the time uh, they are in need. What is Operon? An Operon is a group of genes that are transcribed at the same time. They usually control an important biochemical process. They are only found in prokaryotes. The Lac Operon. The Lac Operon consists of three genes, each involved in processing the sugar lactose. One of them is the gene for the enzyme beta-galactosidase. This enzyme hydrolyzes lactose, which is a disaccharide, into monosaccharide called glucose and galactose. Adapting to the environment. Ecoli can either use glucose, which is a monosaccharide, or lactose, which is a disaccharide. However, lactose needs to be hydrolyzed first. So the bacterium prefers to use glucose when it can. Operon model. So you can see in the diagram P that forms the promoter, I is the inducer, O is the operator region, Z is the gene that codes for beta-galactosidase, Y the gene that codes for an enzyme permease, A the gene that codes for transacetylase. The genes are represented in yellow color, which forms the structural genes. So it is the I region, that is a PIPO region that forms mRNA in the presence of inducer. So this repressor-inducer complex does not bind DNA. Once this repressor-inducer complex does not bind, the genes, the structural genes will be transcribed to form mRNA, and transcription genes are switched on. Designation of gene. LAC-Z that codes for an enzyme beta-galactosidase. The function of LAC-Z is to break down lactose into glucose and galactose. LAC-Y gene that codes for an enzyme galactose permease. This protein found in the Ecoli cytoplasmic membrane actively transports lactose into the cell. LAC-A gene that codes for an enzyme thiogalactoside transacetylase. The function of this enzyme is not known. It is coded for by the gene LAC-A. Four situations are possible. When glucose is present and lactose is absent, the E. coli does not produce beta-galactosidase. When glucose is present and lactose is present, the E. coli does not produce beta-galactosidase. When glucose is absent and lactose is absent, the E. coli does not produce beta-galactosidase. When glucose is absent and lactose is present, the E. coli does not produce beta-galactosidase. The control of the Lac Operon. When lactose is absent, so this is a situation they try to give. So in the absence of lactose, a repressor protein is continuously synthesized. It sits on the sequence of DNA just in front of the Lac Operon, the operator site. The repressor protein blocks the promoter site where the RNA polymerase settles before it starts transcribing. So the diagram clearly reveals that the repressor protein binds to the operator and thereby it represses the promoter site where it initiates transcription by RNA polymerase. In situation 2, when lactose is present, a small amount of sugar, allolactose, is formed within the bacterial cell. This fits into the repressor protein at another active site that is called allosteric site. This causes the repressor protein to change its shape. That is, it brings a conformational change. It can no longer sit on the operator site. So RNA polymerase can now reach its promoter site. So in the second situation, it shows that the repressor protein binds to the allolactose and sits on the allosteric site. Thereby, it brings the conformational change. Now, this repressor protein along with this allolactose can no longer sit on the operator site so that RNA polymerase transcribes the structural genes. When lactose is present, a small amount of the third situation, when both glucose and lactose are present. This explains how the Lac Operon is transcribed only when lactose is present. But this doesn't explain why the Operon is not transcribed when both glucose and lactose are present. Actually, when glucose and lactose are present, E. coli first prefers the utilization of glucose rather than digesting the lactose. The reason is not known why this happens. When glucose and lactose are present, RNA polymerase can sit on the promoter site, but it is unstable and it keeps falling off, thereby no transcription occurs. Right? E. coli simply utilizes glucose. The fourth situation, when glucose is absent and lactose is present, another protein is needed, an activator protein. This stabilizes RNA polymerases. The activator protein only works when glucose is absent. In this way, E. coli only makes enzymes to metabolize other sugars in the absence of glucose. So this is a summary what we had seen earlier. Carbohydrate, both glucose and lactose present. When you look into the activator protein, it doesn't bound to DNA, whereas repressor protein is lifted off from the operator site when both glucose and lactose present. RNA polymerase, it keeps falling off promoter, so Lac Operon, no transcription. So in the second situation, when glucose is present, lactose is absent. When you look into the activator protein, it doesn't bound to DNA, whereas repressor protein bound to operator site. RNA polymerase is blocked by the repressor, no transcription occurs. In situation 3, glucose and lactose both are absent. Activator protein binds to DNA. Repressor protein binds to operator site. RNA polymerase is blocked by the repressor, no transcription occurs. Fourth situation, when both glucose and lactose present, activator protein binds to DNA. Repressor protein is lifted off from the operator site. RNA polymerase sits on the promoter site. There is a transcription occur. Thank you.